In this video, I will discuss how DNA codes for amino acids that are assembled into proteins. We know DNA stores its information in a sequence of nucleotides. The nucleotides have three parts: a sugar, a phosphate, and a nucleobase. It is the nucleobase that distinguishes nucleotides from each other. There are 5 nucleobases: adenine A, guanine G, cytosine C, thymine T, and uracil U. A GCT are the DNA bases. A GCU are the RNA bases. Adenine A pairs with thymine T in DNA, or with uracil U in RNA. Guanine G pairs with cytosine C. So, we get a T and GC pairs in DNA. We get a U and GC pairs in RNA. We know that mRNA is transcribed from DNA, and that mRNA transports the protein coding information to the ribosome. So, how does the ribosome translate the mRNA code into proteins? How do we read the AGCU RNA code? To start, the protein code is written in codon triplets. Each codon triplet is 3 letters of the AGCU nucleotide code. Every 3-letter codon sequence is a marker for a particular amino acid, a start marker, or a stop marker. The code transcribed upon the mRNA is easy to read if you have a codon table. The codon table is 8 by 8 for a total of 64 codons. 61 codons code for amino acids. 3 codons, UAA, UAG, and UGA, are stop codons. These stop codons tell the ribosome to stop. The protein is complete. The codon AUG codes for both methionine and tells the ribosome to start synthesizing protein. A codon table is shown. There are only 20 amino acids that are regularly used in eukaryote cells. These are the 20 canon amino acids. There are 3 stop codons and 61 permutations of the 3-letter codons, so most of the 20 amino acids are coded for by more than one codon triplet code. UUA, UUG, CUU, CUA, CUC, and CUG are all codons for the amino acid leucine. Tryptophan has only one codon, UGG. The only other amino acid with one codon is methionine, which is coded by a UG. The codon AUG is also the start codon. An amino acid is any acid with an amino group NH3 and a carboxyl group COOH. There are over 200 known amino acids, and probably a lot more chemicals that could be amino acids. There are only 20 canon amino acids that are coded for in eukaryote RNA. There are 2 extra amino acids that might be found in organisms: selenocysteine and pyrrolysine. Pyrrolysine is found only in methanogenic archaea and bacteria. Selenocysteine can be found in human proteins. Selenocysteine will be added to a eukaryote protein by the UGA stop codon only in special cases, where the messenger RNA contains a selenocysteine insertion sequence in the 3' tail of the mRNA. A question that no one has the answer to is, of the hundreds of amino acids, why are only 20 or 22 used by organisms? Some evolutionists believe the earliest life could have started using as few as 10 amino acids, and then evolved to use the 20 canon amino acids. The evolution from 10 to 20 amino acids must have occurred fairly quickly, as the 20 amino acids is standard for all life forms, with the few exceptions that can use the 20 canon plus the 2 non-canon amino acids. Why stop at 20 or 22? Why didn't some evolutionary lines go to 30 or 40 amino acids? Some biologists contend that having multiple codons for the same amino acid guards against mutations. So why wouldn't codon quadruplets be safer for multiple redundancy than codon triplets? Fazale Rana answers some of these questions in his article Why These 20 Amino Acids at the link shown. A short answer is 20 amino acids for 64 codons is optimum. Life is about optimization, not too simple, not too complex. In very complex systems, some aspects of the system must be left suboptimal so that other parts of the system can be made better. Francis Crick proposed the frozen accident hypothesis at the NIH link shown. Crick proposes evolution had come to use the 20 canon amino acids by the time of the last universal common ancestor, Luca. And evolution got frozen at the 20 canon amino acids by accident. For more details on the frozen accident hypothesis, see the Kunin and Novozhelov paper at the NIH website shown. There is more to discover about life and amino acids. There will be more discoveries. There are some things that may never be known. There are limits to what can be known. In 1953, the physicist-mathematician George Gamow predicted that 20 canon amino acids would be coded for by 3-letter codons. He knew that RNA had 4 nucleobases, a GCU. He knew that a 2-letter codon could yield only 4 squared or 16 permutations. 16 permutations are too few to code for all 20 canon amino acids. A 4-letter codon would yield 4 to the 4th power, or 256 permutations. 256 permutations would be too wasteful to code 20 amino acids. A 3-letter codon yields 4 to the 3rd power, or 64 permutations of codons. A 3-letter codon would be able to code for all 20 canon amino acids, with a few repeats plus a few special instruction codons like the starts and stop codons. 1953 was the same year that James Watson and Francis Crick discovered the double helix structure of DNA. They did so with the help of Rosalind Franklin and Maurice Wilkins. In 1961, Francis Crick, Sidney Brenner, Leslie Barnett, and R.J. Watts-Tobin conducted the Crick, Brenner, Barnett, and Watts-Tobin experiment that proved that the codon was 3 nucleobases long, just as George Gamow had predicted. The Crick et al. Experiment demonstrated that when sets of 3 nucleobases are removed at a time from RNA of bacteria, the RNA would still yield usable proteins. However, removal of sets of 1, 2, 4, or 5 nucleobases would frameshift the translation so badly that no functional protein would result. Later in 1961, Marshall Nierenberg and Heinrich J. Mattei conducted further experiments that began to show which codons code for which amino acids. That amino acid decoding process is now complete, and so we have codon tables such as the table shown. Although both the Crick et al. And the Nierenberg-Mattei teams used bacteria for their experiments, it has since been proved that the RNA code for amino acids is universal to all life on Earth. The amino acid code points to a universal common ancestor of all life. If we ever find life on other planets, it would be curious if they make protein with RNA, and highly improbable that their amino acid code would be the same as life on Earth. A believer in intelligent design could turn universality of the amino acid code on its head. It could be just as true that God likes to build with orderly building blocks like universal codons. An intelligent designer standardizes the basic parts of construction. At least in America, all homes are built of concrete, bricks, 2x4 dimensional lumber, and plywood. We chose these materials to keep building tidy, and these are the types of building materials that suppliers keep in stock. It is not because there are no known alternatives. Before the supply chain was invented, houses were custom built from whatever rocks and trees were available at the home site. So, 2x4s are universal because of an intelligent decision by an agreement of the largest suppliers. It is a convenience to use standardized parts, not a necessity. In the world of physics, all matter is composed of atoms which in turn are built of universal building blocks of electrons, protons, and neutrons, or the universal building blocks of the standard model of physics, which are quarks, leptons, and bosons. God chose these natural building blocks to build the universe as a conscious decision, not out of necessity. God might have chosen to use different size protons to build different atoms. God might have chosen to eliminate neutrons and neutrinos. We discover the laws of physics are based on what God decided to do, not what God needed to do. The DNA codon language could just as well be the Word of God, and the genome sequence for each organism is a book of God's words written specifically for each species. If we have questions as to why some genomes are so corrupted with junk, we need look no further than the fall from Eden. Believers have no proof this is true. Atheists cannot prove it is false. To further infuriate both Bible believers and confirmed atheists would be to propose a superintelligence that set up the laws of biology that would allow evolution. In such a proposal, a founder species, Luca, could evolve into the diversity of existent life forms. The proposal of a superintelligence who designed evolution would be dismissed by Bible believers, as the Bible says nothing about evolution, one creature evolving into another kind of creature. The proposal of a superintelligent being that created evolution would infuriate confirmed atheists because the proposal is unscientific. There might be no way to test the proposal scientifically. All scientific experiments would merely confirm the laws of biology are natural, just as the superintelligence designed those laws to be, without ever revealing any knowledge about whether the superintelligent being exists or does not exist. Such problems arise in other untestable proposals such as the existence of parallel universes or the multiverse or other many worlds proposals. So far, all many worlds proposals are untestable, and so serious scientists rightfully claim that they are not scientific proposals. In the same vein, a proposal of a superintelligence who designed evolution would be dismissed by serious scientists on the grounds that it is not a testable proposal. Enough with my tangential speculation on the existence of God. You can end the video now or watch my next video on genes and the genome. Or you can continue this video to watch my bonus feature, where I play around with the mathematics of codons. Bonus: RNA coding by the numbers. The RNA code is a base 4 system or quaternary system, a CGU. Most computer languages used a base 2 binary system, zeros and ones, or off and on, or a yes or no code. If A equals 0, C equals 1, G equals 2, and U equals 3, we can make a table comparing decimal numbers to binary numbers to base 4 and to a 3-letter RNA codon and a 2-letter RNA codon. In the table, we see 9 columns of number or letters comparing base 10 to base 2, base 4, the 3-letter RNA codons, and the 2-letter RNA codons. In column 1, the base 10 or decimal system we are all familiar with lists the numbers 0 through 32. In column 6, the base 10 lists the numbers 32 through 64. I deliberately repeated 32 twice to make a point about base 2 and base 4 ending in 1 followed by zeros. We should note that the base 10 system uses the characters 0 through 9, and then adds a character, x, followed by a 0 every 10th number, 10, 20, 30, 40, 50, and 60. If we went past 99, we will have used up all the 2-digit permutations. The next number after 99 would be x, followed by 2 zeros, 100 or 100. In the base 2 binary system, the only characters we can work with are 0 and 1. Every time we get to a power of 2, 2 to the x power, then we have some form of 1, 0, 0. Examples shown include 2 power of 1 equals 1, 0, 2 to the power of 2 equals 1, 0, 0, 2 to the power of 3 equals 1, 0, 0, 0, 2 to the power of 4 is written 1 and 4 zeros. 2 to the power of 5 is 1 with 5 zeros, the decimal number 32, and 2 to the power of 6 is written 1 with 6 zeros, the decimal number 64. We run out of characters fast in base 2 and need to add character places. Base 2 is not very efficient at keeping the number character places low. In the base 4 quaternary system, I was able to display all the numbers up to 63 with only 3 character places with all the permutations the characters 0, 1, 2, and 3. At 64, I had to add a 4th character place and got one with 3 zeros after 333. Just as in the base 2 and base 10, all the powers of 4 to the power of x, end in all zeros. Examples are 4 to the power of 1 is written 10. 4 to the power of 2 is written 100. And 4 to the power of 3 is 1 with 3 zeros, the decimal 64. I used 0 as prefix placeholders for base 4 numbers below 16, or base 4100, so that we can compare the permutations of the base 4 quaternary system to permutations of the 3-letter RNA codon. Incidentally, I use the word permutation instead of combination to stress the order of the characters is important. 010 and 1000 are the same combination of zeros and ones, but they are distinct permutations of zeros and ones. Permutation or order of the nucleobases is more important to the ribosome than the combination. In the codon code, the permutation AUG means methionine or start, whereas GUA means valine. When I use 3 placeholders for the permutations of the base 4 characters 0, 1, 2, and 3, there is a 1-to-1 correspondence with the 3-letter RNA codons. If A equals 0, C equals 1, G equals 2, U equals 3, then for any 3-number permutation of the numbers 0, 1, 2, and 3, I can find a corresponding 3-letter RNA codon. For example, the base 4, 032 corresponds to a UG in the 3-letter RNA codon code. Incidentally, some computational geneticists have experimented with embedding coded messages into the DNA code of bacteria using a base 4 coding system that can be converted back into binary or ASCII messages when read in a DNA scanner. When we compare the 3-letter RNA code to the 2-letter code, we see the obvious deficiency we would run into if we tried to account for the 20 canon amino acids with the 2-letter codon system. If RNA had a 2-letter codon, we would see that the 2-letter RNA code runs out of permutations at decimal 15, while the 3-letter RNA codon can go all the way to decimal 63 without a repeat. This advantage of the 3-letter codon system for being able to account for all 20 canon amino acids may be why life settled on the 3-letter codon system. We will never know when or in what circumstance the first ribosome gained the ability to read the first messenger RNA that used the 3-letter codon system to produce a protein chain.
Thank you for watching. If you enjoyed this video and want to help me make similar content, please like and subscribe.